The overall goal of this procedure is to use real-time characterization to identify the optimal sonication conditions, intensity, and duration for obtaining stable and uniform nanomaterial dispersions in aqueous media.
This method can help answer key questions in nanoscience, particularly in the field of nanotoxicology, about how to optimize nanoparticle dispersion without impacting sample integrity. The main advantage of this optimization strategy is that it improves control over the final dispersion quality, which is instrumental in ensuring repeatability.
To begin the procedure, calibrate the probe sonicator fitted with a vial block sonitrode. Then, use a clean metal spatula to measure 2 mg of the chosen nanopowder into each of 3 clean 10 or 20 ml glass vials numbered 1 through 3. Pipette 1 ml of deionized water along the inner walls of each vial. For hydrophobic samples, instead use 1 ml of 0.5% by volume ethanol in deionized water. Mix each sample into a thick paste. Then, add sufficient deionized water to the paste to achieve a final sample concentration of 0.2 mg per milliliter. Horizontally swirl the vials to dislodge any nanopowder adhering to the inner walls. Transfer 1.5 ml of each nanopowder dispersion to clean microcentrifuge tubes labeled with the corresponding numbers. Firmly close the tubes and swirl them to dislodge nanopowder from inner walls. Then, place the samples in